Okay. Thank you very much for joining, all of you. So yesterday we saw applications of bioinformatics mainly. I'll just give a quick revision of yesterday. Yes. So yesterday we saw what is bioinformatics, what are applications of bioinformatics, what are the major bioinformatics skill areas, which are divided into 3 categories: structural bioinformatics, functional bioinformatics, and sequence analysis of bioinformatics. And I used few databases to explain these skill areas. For sequence analysis, I used NCBI, and for structural analysis, I used the database like PDB for protein structures visualizing, and for functional analysis, I used string database, which is used to understand the protein-protein interactions, as well as the another database I used was HPRD, Human Proteome Reference Database. So today we will see ahead that about databases. Actually, what is database? Because whichever information we use and store and retrieve throughout the internet, we need database. So before starting our course, we need to store these. In order to analyze, we need to understand that what is a database basically. So today I will start the overview of major bioinformatics databases. So first of all, let us what is a database. So today I will cover what is a database, types of databases, what is the need of a biological database, and the classification of various bioinformatics databases. I will list the names, and which are the major bioinformatics databases. I will give an introduction to the major bioinformatics database, because I think that this much will take the time and will take question answer also. So I would not be able to cover today the whole description about information retrieval from these biological databases. And so up till here, what is database, types of databases, need of a biological database, and bioinformatics database classification, and names of the major bioinformatics databases I'll introduce. Up till here I'll cover. So about what is a database. So actually, what is the function of a database? As the name suggests, database is actually a structured collection of information pieces. Whatever information you get from anywhere, that is the raw data. It needs to be stored somewhere, right? So it needs to be stored somewhere which can present that information in the meaningful way. And those information pieces must be related to each other, or they must refer to a common specific point, that what they are referring about. So this is the basic function of a database, that it presents the information pieces into a structured collection, into a structure, and these information pieces are related to each other. They refer to a common point. So how we can understand it? So for example, roughly we take the examples that in a school, the students are there. The students in a school. School is a database and students are information pieces. Again, library of books. Books is the database to store library is the database to store books. Similarly, bank which stores money. So this is a basic idea about database, how to understand. Now, similarly, this I was talking about any piece of information we keep. So, what is the need of a biological database, and what is a biological database? So, in biological databases, we generally talk about the biological information like DNA, RNA, macromolecules, taxonomy, nomenclature. Many things we come across every day in the biological experiments. So, where to store them? So, that can be done through a biological database. So any information which needs to be stored at a place from where it can be accessed meaningfully and can be distributed easily, we need a database. And for bioinformatics, large amount of sequence data is there. Sequence data means sequences of the DNA, RNA, proteins, and their structured collection in the terms of then we need to understand its functional information, its functional areas, and every day this data comes to us. So we need the bioinformatics database, and here comes the need of the biological database or the bioinformatics databases. Now we will take few examples through these figures, and we will see that what are the databases, how the information is stored in database. For example, students report card is an example of database, as you can see in the figure. In one column, the subject has been mentioned, and in the other columns, the term-wise marks have been mentioned, how they are arranged and how they are related to each other. They are referring to the report card, the common specific point students report card. So this report card is working as a database, and the information stored are arranged in a structured form. They are working as the pieces of information. So we can see it, that this is a database example. Secondly, library, as I told. In library, for example, number of students are there, which student has borrowed which book. So the one column is for borrows, one column is for students, one column in database management system it is mentioned as books, one column is for authors, types. So this is the example of library as a database in database management system. Now, basically, through these examples, we understood the databases. Now we will understand what are types of databases. We can understand the classification actually. So first comes hierarchical database. So what is a hierarchical database? So in a hierarchical database, when there is a single root node and other data which is linked to that root node are linked to the same root node, and they are expanding like a tree. Means there is a root node and the linked data are going in branches downwards. We will see the figure. This is the hierarchical model of database. For example, there is a college and there are several departments, bio department, chemistry department, other departments are there. So in each and every department, there are courses. Wait for a moment, the figure for hierarchical model is not presented. Just wait. Can you see the figure of hierarchical model of in database management system? Okay, yes, it came. So in a college, you can see in the figure, there are various departments. So for example, there is chemistry department, biology department, other departments, physics department, and in every department there are courses. For example, biology, life sciences department is there, and the courses are bioinformatics, microbiology, biotechnology, medical biotechnology, and other courses are there. Then again, teachers are there for each course, students are there for each the departments. So again, the department has been subdivided into course, teachers, and students. Students will be again divided according to the name of the course. When teachers will be divided accordingly, and the course will be divided. Course will be divided like in theory and labs. So basically, we can see that hierarchy is taking place. If there is a root node for college, and the other linked data are expanding in a tree-like figure. We can see that this is the hierarchical model in a database management system. Now the second type of database is network database, network type of database. So here we can see that there is a multiple relationship with each of the data linked. Means if there is one, there are pieces of information of one data, then all of them are linked multiply with each other. And this makes the network database very flexible database. How we can understand it through this figure. For example, there is a store, and in a store we can see that in a store customer also comes to purchase, manager also is there to manage the store, and salesman also is to sell. So all are from the same store, and all of them have relationship with each other. How? Through the order. Through the order, how? Because customer purchases the order and gives the order to purchase, manager then releases the order, and salesman sells the order. And similarly with the items also. Items list is given, and so customer, manager, salesman are related to each other. So it is making a network, and it is increasing the flexibility of storing this information pieces to make it more flexible arrangement. So this is network database model in database management system. Then comes relational database. So in relational database, what happens? It uses the relationship of similar type of data in the in columns, and we can compare various data entries due to the presence of columns. So we can compare also here. So we can see that this is an example of relational database model. So here the citizens of a city are given, in which the names of the citizen are given in one column, then a title is given, name is given, city is given, age is given, and salary is given. And how we can, they belong to the same city, and we can compare that, okay, this is the name of the person, this is the name of their, how many persons are in this city, what is their age, and what is their salary. We can compare also, though they are having relationship between, and they are presented in not hierarchical like network database. So it makes yes, some question is coming. Can you write in the chat box, please? Just a second. We can take the question answer at the last session. A sound problem. Okay. Now you can hear my voice. Can you hear me? Okay. So I'll present it again. Yes. So I presented hierarchical model in database management system. Was it audible to you? Okay. And network database model was audible? Last relational database. So I will explain it again. So in relational database model, the various pieces of information are presented in the form of tables. And each of the data pieces of information are related to each other, and we can compare. For example, if the names are given in one column, then another names are given in another column, and then city has been given, in which city they live. Then the age has been given, the salary has been given. So they have been arranged in columns, and each entry, for example, for Smith, John Smith, the city number is 3, and his age is 35, and his salary is $280. So we can compare this entry of John Smith with Jane Doe, the second entry, that he resides in city 1 and he has age 28 and he has this much salary, 325. So we can compare the pieces of information by arranging them in column form. So actually in relational database model system, it becomes more flexible for us to compare the data. But in hierarchy database model and in network database model, it is not easy to compare the pieces of information, because the pieces of information is not arranged in columns like this. Now we come to object-oriented database. Actually, what is object and what is to understand object, we will understand something called class. What is the class? So actually, object comes under class. So we can understand this as if fruit is a class, then what are the objects? If fruit is a class, then objects are mango, banana, papaya, lychee. These are the class. These are the objects. So class is basically a wider area, and the objects are basically the subterminals under the class. So this object-oriented database basically makes the classification of objects in various classes, and these classes are arranged in columns. So here we see that I have written that this database model contains data in the form of separate columns. It means the classes are arranged in separate columns, and each column contains a specific class of objects. Means objects of one class will be in one column, objects of second class will be in second column, and they are put in another different tables. Not in a single table, they all are put. In a relational database, we saw that the entries were put in the single table in rows. But in object-oriented database, we don't see. We will see this in the figure. Yes. So in relational modeling, we see that employee table, department table, and job table, all the three are arranged in the same table as the three columns. But in object-oriented modeling, object-oriented database, what is done, that employee is made as a class. And in employee, the manager, the secretary, and the clerk will be made as subclasses in one table. Again, the department will be made as one class. And according to it, the department of the manager, the department of the secretary, and clerk will be their tables and theirs. So this way we can see that the classification is object-oriented based on the objects which are present in a class. They are further classified. So we saw the types of database. Now we saw through figures, we understood the hierarchical model in database management system, the network database model in database management system, the relational database model in database management system, and we saw object-oriented database model in database management system. Why we saw these types of databases? Because we will see their uses in biological databases as well. That how the biological databases use these data to keep the information about various macromolecules, biological macromolecules. So here comes the need of a biological database. So actually what is happening, that biological information is increasing very, very fast. So we are having this data, biological data earlier, as I told, that in the forms of genes, proteins, their sequences, their functional, their structures, their classification, their annotation, their nomenclature, taxonomy, information about their NMR experiments in the form of protein-protein interactions, and the data obtained from it. Means there is a lot of data to store. So the biological database comes into picture. And if we store the biological, we make the use of biological database to store this information in a proper format, then we can make the use of this information for our further understanding, for its further classification, for its further relationship between the various data, to make its use in research. So these are the data types basically in biological database. So first is, as you can see, it is primary data. So what is primary data? Primary data is basically in the form of raw sequences from a primary database. So actually primary database contains the sequences, basically either the sequences are DNA sequences, or in a protein they are either in the form of amino acid sequences. So these are the primary databases, and the function of the primary database is to store these sequences or these primary data. Then comes another type of data which is stored in the biological database, is known as secondary data. So this secondary data is actually in the form of motifs, in the form of regular expressions, in the form of blocks, profiles, fingerprints. So this is called secondary protein structure information. So it is in the form of protein structure, but it is secondary, and that is stored in secondary database. And then comes tertiary data, which is stored by tertiary database. And this tertiary data is actually the three-dimensional structure of protein. Then there comes atomic coordinates, which are basically present between domains, folding motifs, etc. These are also the data. And another one more type of data is specifically about organisms which are stored by specialized databases. For example, a fly base is a database which stored a worm base, fly base, which are organism specific databases, and they are known as specialized databases. Now I have classified the bioinformatics databases. They are classified into following and the basis of their content. So at present, biological databases use three types of database structures, including flat files. Actually, flat files means the information is contained within the database with delimiters, that is a standing line, and the raw information is only there in simply. One of the example is GenBank. In GenBank it is present, sequences. Then biological databases also use relational format of model. Then they also use object oriented. Now we do not need to understand right now that how this relational object oriented, how genes are put in relational, how they are put in object oriented. We will first understand that on the basis of content, what are primary biological databases, what are secondary biological databases, and what are specialized databases. Of course, we will see the formats in which these biological information is stored, but before that we will know that which databases use which type of content. So primary database, as I told, is use primary data. This I told primary data, which is sequences, basically raw sequences in the form of DNA and amino acid for proteins. So they store raw nucleic acid sequence data, and basically it is ACTG bases, nucleic acid bases. Here you can see in the figure, this is primary data, AATC. This is the in the form of this sequence it will be present in primary databases. So in the form of raw nucleic acid sequence data produced and submitted by researchers worldwide. So worldwide the researchers submit, first they do wet lab experiments and they produce and submit the data into primary databases. So the examples of such databases are, primary databases are GenBank, PDB, DDBJ, EMBL, etc. Okay. Then comes secondary database. Secondary database stores secondary protein structure information. So in the form of motifs, we can understand what are regular expressions, blocks, profiles, fingerprints from the secondary protein structure. And that is stored in secondary databases. So this way they help in adding more biological information to the raw sequence. So first a raw sequence is obtained, and what is the function of this raw sequence, I mean, how can we analyze it? How can we understand that, okay, this is the gene region, this is the promoter region? How can we predict it? That can be done through secondary database. And the examples are Swiss prot, translated EMBL, that is TR-EMBL, PIR, that is protein information resource, etc. Then comes specialized databases. As I told you, here the focus is on specific organisms. Organism-wise, it stores the data for a specific research community. Example is ACE-DB, ACE database, which is a microbial database. Then TAIR, Flybase, etc. TAIR is also example of genome database. So they basically contain the organism-specific genomes like that. Okay. Now you can see this figure, the classification of database. So database has been classified into two types of databases, basically on the basis of the data nucleotide sequences and protein sequences. So nucleotide sequences basically are stored in primary, secondary and tertiary databases, as I explained now. So which are the primary databases? EMBL, European Molecular Biology Laboratory, GenBank and DDBJ, DNA Data Bank of Japan. So these three, EMBL, GenBank and DDBJ, store primary sequence data of nucleotides. And what happens in protein? In protein, actually there is sequence, there is interaction and there is structure. So sequence is stored by UniProt database, PIR, which is Protein Information Resource Database, and Swiss prot database. These are primary databases in protein. And again, interaction database means to understand the interaction between one protein, another protein, or multiple proteins. There are databases like BioGrid, String. I will give you this lecture in which you can memorize or you can keep this in mind of this classification. So it will be very clear to you about the classification of databases. And the third one is structure. So the third classification is structure database. On the basis of structure, we can classify the database as PDB, Protein Data Bank, CAP, SCOPE, etc. Here, here SCOP should be there. E is extra. So CAP, PDB stands for Protein Data Bank. CAP stands for Class, Architecture, Topology, Homology, and SCOPE stands for Structural Classification of Proteins. So these are the structural databases which identify the structure, three-dimensional structure of the protein. So and the third classification is about specialized databases. One of the example is OMIM, which is Online Mendelian Inheritance in Man, which keeps the information, stores the information about inherited diseases. So these are the examples of the classification of databases. Now, which are the major bioinformatics databases? So the major primary bioinformatics databases which contain primary sources of data like nucleic acid sequences, protein sequences, structural information of biological molecules, etc. are NCBI, GenBank, DDBJ, Swiss prot, PIR, PDB. So here I have taken this nucleic acid sequences, NCBI, GenBank, DDBJ, as well as I have taken Swiss prot, PIR and PDB for protein sequences. Okay, and about which are the major secondary bioinformatics databases. So these are the databases which contain additional information in addition to that derived from the primary database, means they add information to the sequences, as I explained earlier. So secondary databases can be analyzed in a variety of ways and they have different formats. So here the secondary databases, major secondary databases are translated EMBL, TFAM, Protein Family Database, ProCite, Profiles, Structural Classification of Proteins, that is SCOPE, and CAT, Class, Architecture, Topology and Homology. So now here I have written all the major bioinformatics databases together: NCBI, GenBank, DDBJ, Swiss prot, PIR, PDB, translated EMBL, PFAM, ProCite, Profiles, SCOPE and CAT. So today we will see one by one we will take each of the database now, and we will see that how to operate that database at a basic level. How we can use, how can we make the use of these databases at a basic level to start with. So first of all, let us take NCBI, which is very popular database of bioinformatics. Most of you all be knowing about how to use. Still, we will start from the very basic level. So we will take NCBI. So actually, NCBI is the National Center for Biotechnology Information. We will go to Google, we will type either NCBI in the search box, or we can type here ncbi.nlf.nih.gov. So here NCBI stands for National Center for Biotechnology Information. NLM stands for National National Institute of Health, and NIH stands for National Institute of Health, and NLM stands for National Library of Medicine. GOV stands for Government Organization. So this is the repository of databases, several databases. We go to NCBI, and it is a collection of many databases, several databases which we can use in order to understand and access the biomedical and genomic information. So we open the home page of NCBI, and here we see that here it is in the search box, it has been written all databases. Here it means, and here we can search the various databases. Which are the databases in the scroll? Here we can see assembly, bio collections, bio project, bio sample, bio systems. So many databases are there. So we can search each of the databases. So I will discuss the main things which we use in generally in research projects, which come across very frequently for us. So like protein database is there, PMC, which stands for PubMed Central, which is a literature database, is there. OMIMI tool, that is online Mendelian inheritance of man, which is tells the information about the diseases. Nucleotide database stores nucleotide sequences. Then PubChem is there, database for chemical compounds, protein clusters. Proteins are stored in the form of clusters, in the form of protein databases there. Then SNP information about single nucleotide polymorphism is stored here. So it is a collection of many databases. Now, for example, our purpose, let us start with a purpose to how to visualize any protein or nucleotide sequence. So let us take a protein, any protein we can take. For example, we take human prion protein. Okay. So we will type here all databases, or we can select we want to see the protein sequence. So we can select, we can either see the nucleotide sequence or we can select the protein sequence. It depends upon us. Yes. Abrar, your question is factor XA. Okay. So what do you want to ask? Are you asking from NCBI? Yes, yes. You need to search factor search bar. Okay, you want to, this is, what is XA, Abra? May I know? Is it a gene or a protein? You need to select search for biological information. Hello? Yes, protein. So let us search for XA. Factor, okay, you want to know about gene factor. So that will come, you will have to type the name of the protein in the search box and in all databases, and for example, if you search for XA, then the result will come for all the databases. Yes. For example, it is a blood coagulation protein. So if you search here, simply we do not need to write the whole here. See, you can see here proteins. in this protein, the protein, this much number of proteins. You want to be very specific. If you want to be very specific about blood coagulation protein, so we will have to type the name also. Let us see in proteins. Okay. So, for example, we are searching for XA protein, which is a blood coagulation protein, as Abrar told. Factor XA is a blood coagulation protein, and we want to obtain information about it from very basic level. So we saw that about actually this much number of 46,869 number of entries have been deposited every by different people. And these are the source databases. In the left side, the names of the species has been given. In animals, this much 32,504 sequences of XC has been deposited, of plants this much. So now the question arises that if we want to search about particular specific protein XA, then we will have to search. We should know whether that what is the source of the data, what is the source database. For example, is it a PDB? Is it a RefSeq? Is it a Unicode knowledge base? And what are its genetic compartments? So since it is a blood coagulation protein, so we will search in yes, it is chloroplast, mitochondrion, plasmid, plasmid. We will search in Homo sapiens also. It is a blood coagulation protein. So for Homo sapiens, we will find out the result. Here the first result has been given. We will click on it. And before searching for it, just a moment, I'll go back. I have to explain something here. So here, Axi Homo sapiens, here it has been given that this protein is 311 amino acids long, and there are two accession numbers. This is accession number, and this is the gene ID. This is the gene ID for this very protein. And there are these are the formats in which we can visualize this very protein sequence. So we want to obtain the detailed information about. So we will click on XA Homo sapiens. So Abrar, did you get why we search for Homo sapiens? Because we are knowing that XA is a blood coagulation protein, so we search for Homo sapiens, and we search for protein database because it is already protein. This is very simple thing. You would have it would have been clear to you. Okay. So now in XA Homo sapiens, this is the result. This came to us. So this is GenPept. This is actually the format in which this very protein has been shown to us. We'll scroll a little and we will go this. This is the whole GenPept format. So actually, format of any protein in which a protein is displayed can be chosen from here. These are the various formats. So we will first analyze the GenPept format because it gives the detailed summary of this proteins which we select. So here the GenBank accession number is given of this very protein. This is the GenBank accession number. It means that this protein has been taken from GenBank. This GenBank, DDBJ and EMBL submit their sequences, they exchange their sequences to NCBI on a daily basis. Also, they exchange their sequences among each other on a daily basis. So from there, this sequence has been taken, and there its accession number is this much. So for example, if any accession number is given to you and you do not know that which protein it is, then you can find it through NCBI. For example, I give you a problem of accession number and I tell you that okay, I want the name of this protein. Tell me. Then what you will do? You will have to find out that which accession number is this. Whether it is a GenBank. If it is a GenBank, it will be in this format. It will be 3-letter code, and then it will be number and decimal. This is the actually released dot 2 stands for release. This is the accession number, and this from GenBank it has been retrieved. So now we come to, this is the list of identical proteins. This is FASTA format and this is graphics. When we will click on identical proteins, we will get the proteins which are identical to this axis proteins. This is FASTA format, which we use very often, and we from this FASTA sequence format also we can retrieve the protein. If we download it, that I will tell you here. This is the locus of locus. In the locus section, it has been given that 311 is the amino acids, is the length of this very protein axis, and it is present in Homo sapiens. These are the accession number. Then this is the database source. So database GenBank. This is the source. When we will click on it, it will redirect us to GenBank, where it is this much. This is the entry number. Then the source is Homo sapiens, organism is Homo sapiens, and this is the classification, various families and classes, eukaryota, metazoa, chordata. In these classes also this this is present, this Xa Homo sapiens, as we saw in earlier. Then the reference here, it has been referred for the since it is 311 amino acids, only the chain has been shown here for 1 to 311 amino acids. Then authors who have submitted this sequence are here. The name has been listed here. Then the title for the paper in which they have released about this sequence has been given here. Mechanism and consequences of the duplication of human. Then the journal is also there. The remark is also that GenBank, here it has been written about that GenBank has submitted this in this article number. Comment is there. And the source are this from first amino acid to 311 amino acids protein. It is this much, this length has been submitted here. So coding region is 50 to 110. Means the amino acid number 50 to amino acid number 110 region is coding for a gene. Functional, it is a promoter region. Then is site. Site type is other. Site is basically the sites where this protein is present in the blood. Then it is cytokine receptor motif, functional domain. This is the functional area. Cytokine receptor motif, it is present. CDD, which is coding domain. This is CDS, coding sequence 1 to 311. And these are the base pair, these are the amino acid base pairs, and this is actually the format is in GenPept. So from 1 to 311, the last is H, and each of the base pairs stands for a particular amino acid. So we have seen now. What we want to do is we have few things like we had accession number. From accession number we can understand that what is the protein. We first search the protein and we saw accession number. If this only accession number is given to us, again we can type it and paste it in NCBI and we can get this very protein. Now we want to save this file. So we can do it from here. So we want to save it, for example, FASTA format is very much used. So we want to save it in FASTA format. So we will click on FASTA. Okay. So this is the FASTA format. This is the accession number. And we want to save this format, for example. So we will select it and we will either send it to file and we can select it in the form of FASTA create file, or we can download it, or we can do that we can copy this and we can paste it in one of the Word doc, any of the doc, and there we can keep it saved. And then we can use that FASTA format later to visualize this protein in in NCBI, or we can use that FASTA format to run BLAST. Here it is run BLAST. BLAST is the basic local alignment search tool, which I will describe later in the software and tools in sequence analysis portion. So we can, this FASTA sequence, the meaning is that this FASTA sequence is want to search any literature in NCBI, we can use that PubMed database or other literature database. Assembly, we can see biocollection. You can explore each and every database one by one and see that what is the use of that database, what does it contain. One by one you can explore, and later in the question answer session we will discuss each of them in details. When we will do projects and practicals, we will include them together as 5 projects and we will include these databases. Then comes GenBank. So GenBank is again the database which contains the nucleotide information, nucleotide sequence information. So we will go to GenBank database. Just a moment. Yes. So for GenBank, we go to GenBank database. We will go to Google and type here GenBank. Okay. So how to use? So this GenBank is actually a database under National Institute of Health. National Institute of Health, and it is an annotated collection of all publicly available. Okay. You can't see. Sound is not coming? No. Sound is not coming. Now is it audible? Okay, from where could you, screen is not changing, okay. Now the sound is audible or not? Okay, okay, okay. So screen is not changing, I'll just see it.
Yeah. Yes.
So I was telling about GenBank database.
Unshare and share screen.
Unshare. How? Okay. Now is the screen visible to you?
GenBank.
Okay. So let us start GenBank. So GenBank also contains nucleotide sequences, only raw nucleotide sequences. The information is added, is not the functional information, it is annotated, but the functional information is not added to it. So it comes under the NIS, which is National Institute of Health, and it is a genetic sequence database. So all the publicly DNA sequences which are available through the various experiments, they are stored in GenBank. So we will see that how this GenBank, how to use basically with how to use this GenBank. Okay. So since I told that it contains nucleotide database, so we can search for nucleotide. You can search here for any nucleotide sequence. For example, again we will take XA factor only. Let us say, is it okay? Okay, XA. So we see the nucleotide. We want to see the nucleotide sequence for XA. So now we are redirected to the nucleotide sequence. Since GenBank is also a type of database, GenBank is a database under NCBI and under National Institute of Health. It is under collaboration with the NCBI. So it redirected us to the NCBI nucleotide page, and we see here Homo sapiens XA gene complete coding sequence. So complete coding sequence has been given here. We can change here from source databases. What do we want? Actually, these databases are the source databases from where this sequence has been collected. So INSDC, so it is the source from where this GenBank keeps its sequences in INSDC. About 5,578 sequences have been deposited here. So let us see this GenBank, this Homo sapiens axis sequence, complete coding sequence. It is 6,873 base pairs. I'll go back. So this is 6,873 base pairs. It is a linear DNA. So this nucleotide sequence is a linear DNA, and its accession number is this accession number of GenBank database, and gene ID is this 250. We see here, we can say here gene ID because actually this is a fragment of DNA. This sequence is not the complete genome. Okay, it is a DNA content. It is a part of a DNA. So it is saying that this is the gene ID, GI. There are so many sequences, about 6,107 fragments have been deposited here. So this much number of sequences, so in genome wise. So when we click on it, we get the complete coding sequence. Means this sequence is coding. This whole nucleotide sequence which has been submitted here is coding. Here down we can see this. This is from the amino acid number 1 to here we can find a coding sequence here. Here above. Yes. So 1 to 6,873 amino acids, and all are coding. All code for all are coding here. So we go bit it down. We see this is the sequence, and this is in the form of this is the GenBank has been taken from the GenBank database. So we want to see its FASTA format. We will click here. This is the FASTA format. You can see in the capital letters. This is in the form of, you can notice here that since it is nucleotide sequence, so it is in the form of G, T, C, A, G, like this. Guanine, adenosine, cytosine, like this. So we go to GenBank, and again here the locus has been given. It is a linear DNA, this much base pairs, as I told you. And this is the accession number. These things you can understand. I described earlier also. Reference, it has been taken this much, 1 to 6,873 amino acids, it has been described. Again, authors and all you can understand, I told earlier. Yes, this is the CDS coding sequence. 2713, 23103, then 3392. These stands for promoter. These genes coding sequences, these are the regions which code for a promoter region. Code 1 starts here, 1. This is the protein ID. It is this sequence is coding for a particular fragment of DNA, and the name of the protein ID and the identification for protein is this. Means this DNA sequence, when is translated to protein, then this is the ID, identification. We will not go into under it because it will we will go further. We are just seeing what is the meaning here. So this much. Okay. Now again we can take the FASTA format from this GenBank database, and we can run BLAST from here to find out the local regions of similarity, identical sequences between this sequence, between XA gene and between the other sequences. We can run BLAST here. We will see how to run BLAST later. Now then comes DDBJ, which stands for DNA Data Bank of Japan. This is also a sequence, contains nucleotide sequence only, and this GenBank, DDBJ, NCBI and EMBL. EMBL is also European Molecular Biology Laboratory. That is also an organization, and these all GenBank, DDBJ and EMBL. I will write here EMBL. Okay. So these all they all share data on a daily basis of the sequences, nucleotide sequences, DNA sequences on a regular basis. Then comes Swiss prot, PIR, PDB. Now we will see about DDBJ. So before saying about DDBJ, up to this, is it clear to you? If I give you any sequence or if I ask you something from up to this, I think you can do it. Is it okay for up till now? Okay. So I want to give you a question. Just you'll type in the chat box. I will do a practical here, and you will have to type in the chat box what to do the next step. So for example, I am a beginner and I have a particular protein. For example, say I have hemoglobin, and I don't know that from where to get the sequence of this hemoglobin, or from where to get the genes of this very protein. Then what we will do? What I will do? I will go to NCBI, and what I will do? Please write in the chat box the next step. After going to NCBI, what I will need to do? What I need to do? Please write in the chat box. I have a protein. Okay. So you are writing that I have to type HB in search bar. Okay, here I have to type HB. For example, I want to search only for genes. Then what I will do? For example, if I type here HB, hemoglobin. Okay. So from here also the genes has been, the list of the genes has this much number of genes. We can go here, or we can particularly, as Abrar told, that we can select from gene database, collection of genes for hemoglobin here. Okay. And we can get the list of genes. Now, if I want to search, I want to see that whether the structure of this hemoglobin is present in this means whether it is present or not, whether it is available or not. Which data source should I select from the left hand side? Please tell me. What I have to do next? I want to search for the protein structure, the data source of the protein structure. What I need to do? Shall I select protein from here? Right? So again, when I select protein for hemoglobin, yes, then I will have to decide the database. Which database shall I select in order to visualize the protein structure? The source data. PDB. Okay. Now, if in PDB, if the data source is not present, PDB is not present, I want to search for only sequence content, then I will search in these. You can see here that these are the sequence content. CCDF, Ensemble, RefSeq, Gen. So these are the databases for sequences. So for PDB I will explain you later, but we want to just see for the sequence right now. So it is okay to you. Now we will go to, shall we, what is the time? Shall I explain right now the next, because it will take time for me for the 3 databases I have taken.
I think you can explain one more.
Okay. Okay. So I will explain DDBJ to you now. DNA Data Bank of Japan, how to use. Okay. So again, we will understand about DDBJ. Yes, yes. Okay. So this DDBJ stands for DNA Data Bank of Japan. So actually it is it comes under National Institute of Health, this also, and it also is an archive of public sequences which are deposited here, and they are freely accessible to us, to the public. Its URL is ddbj.nig.ac.jp. JP stands for Japan. So when we click here, so this is the home page of DDBJ, and we can see that there are 3 options here: Bioinformatics DDBJ websites, annotated assembled sequences, and sequence read archive. So for example, I want to search for what is the meaning of annotated assembled sequences. So here the data which is contained, which is deposited here, is basically annotated. Annotated means adding functional classification, functional information to a particular raw sequence. So it comes under the secondary type of data. So this DDBJ contains the secondary data. So how we can see here, we will go to this search and analysis tab, helps us to search and analyze the DNA sequences. So these are the various search and analysis tools like get entry, which helps in retrieval of the annotated data, means annotated sequences. If any raw sequence, we can search it from here with the help of, if we have accession number. And from where we will get the accession number? We can get it either from NCBI. Up till now we have studied about NCBI only, so I am mentioning here NCBI. Otherwise you can use SwissProt, or you can use GenBank, or you can use this EMBL also you can use. So get entry, ARSA, DRA search, these are the tools pre-publication. So this is basically, we are interested to find out the annotated sequences. How does it look like? Okay, we will go to the home page, and we will again, for example, we search for cancer, annotated sequences for cancer or a disease. We will search here. Not websites. Okay, websites. By mistake I searched websites. Anyways, websites, if we will search websites, we see that all the websites for this which are linked with DDBJ have come about cancer genomics. From here you can get the insight about genomics of cancer, ARSA, DDBJ, ARSA. Let us see. You can see it, you can explore it. The various articles are there, various studies about this. Let us click here. Cancer genomics. Genomics is the study, as I told. Yes. So this is the study. The study has been made. This is the submission date. This is the study title. That whole exome has been analyzed. And this is the study type, cancer genomics. Genomics is the study of the genome. Genome means total DNA content in an organism, in Homo sapiens. So the exome portion, exome analysis has been done. And there are also many. For example, it has been done for myelodysplastic syndrome. And again is biliary tract cancer. Then omics for, so these are 207 studies have been done, and these are the submission dates. You can click on the, you can, what is the basically the purpose here, these are the annotated, you can get the functional information about the study, about the literature about these. So for cancer you can get about, if you want to make a project on exome of a particular protein, first you will get its accession number from NCBI. Then you will get type that accession number, you will get particular protein, you will type in the search box in here the name of that protein, and then you can find out the various studies related to it if you want to know about the annotation portion. Okay, now we want to this was about means annotation portion, and I want to search, this was about the websites, and I want to search for, yes, I want to search for annotated assembled sequences. How do they look? So for cancer, let us see, we can see related to diseases specific. If you have particular, for say you have colorectal cancer, or you have any specific type of cancer, you can search by putting the name of that cancer. If you have COVID-19, you can search also. Let us search for COVID-19. Let us see what comes, whether it has been deposited or not. So websites you will search, then again it will come websites we have searched. Let us search for annotated assembled sequences. How many sequences have been assembled for and this COVID-19? How many sequences have been added with functional information? Okay, so this is the result. 2,164 entries have been found. So these are the name of the primary accession numbers. These are, and here where they have been found. So this is the description, that the severe acute respiratory syndrome coronavirus 2 genome assembly. Yes, look, for chromosome 1 has been done. The sequence length about 29,903 amino acids have been sequenced. Molecular type is RNA. Okay, so you can find it out on the basics of the type of research you have. So you can see that these are the annotated sequences. We will make, certainly we will work on the small projects based on these databases. On everything we learn, we will make a small project and we will work on it. So we saw, yes, and the third thing, again I need to, yes, sequence read archive. Again, let us see. COVID-19. So these are the archived sequences basically, which has been already done and added here. So these are the formats, these are the meta files. From the various sources, it has been collected here in the form of meta file from this Genomics UK Consortium. Submission date has been given, submission COVID in the form of COVID-19. So these all you can search from PubMed of the NCBI also literature database. You can search it from here also. Okay. So roughly we saw NCBI, GenBank and DDBJ today. So up till this, I would like to conclude here about introduction. Later, tomorrow we will see these EMBL, SwissProt, PIR, 3 or 4 5 to 6 databases we will see. So again now I have to make some important announcements we need to make. So up till this, I will show the slides one time more. Yesterday's also I will show. Yesterday's I had sent. Today also I'll send it to you. So we saw what is the database. Okay, structure collection. Recall all these things. Then we saw the various figures. Then we saw the types of database: hierarchical, network, relational, object-oriented. We saw the figures for it. We understood it through examples. Then we saw the need for a biological database, why it is needed, because biological information is increasing very fast, and so on. Then we saw the data types, which are the basic data types in biological data, and where they are stored in primary, secondary, and tertiary databases. We saw the classification of the primary, secondary, and tertiary and specialized database. We saw this diagram for the classification. I will send it to you. And the primary databases we saw, we saw the names: NCBI, GenBank, DDBJ for nucleotide sequences, and for protein sequences, SwissProt, PIR, PDB. And secondary databases as these translated: TREMBL, PFAM, ProCite, Profile, SCOP, and CAT. We will discuss all one by one. NCBI, GenBank, DDBJ, basic how to use them we discussed today. So tomorrow we will discuss the next databases. Up till now, you can explore these databases and you can come up with your question and answers. You can either mail it to me on Bioinformatics videos. So we can take it at the end of the session next week. And now some important announcements need to be made now. So Rajiv sir will make those announcements to you.
Yes.
Okay. Rajiv sir, please.
Hello everyone. Am I audible? Hello? Let me just try it.
Okay, you are audible.
Okay, great. So let me just pull it out. So hope you liked the session, first of all.
Welcome and thank you for choosing free conference calls.
Great. So basically, Faisal, our email ID is bioinformaticsvideos@gmail.com, from which you have already received the meeting invites.
Yes.
So that is there. And basically what I wanted to discuss today was, after this session which Pragya gave, basically she's been really working hard and preparing and trying to add value to everyone's life. But that's she loves doing, she loves biology, bioinformatics, and she loves speaking. So that's the job satisfaction she's aspiring for. But at the same time, whatever efforts we put, we aspire to monetize it as well, because we are putting a lot of efforts there. So job satisfaction ke saath jeeb satisfaction bhi chahiye, that way. So basically, we wanted to share with you all the course which we are offering, which is going to be the aims. So you can see my screen, I guess. Yes.
Course structure.
So that we had shown yesterday, the course structure, and I'll just show the course structure
because some
today as well. Yeah. Yes. So there it is. So typically, I'll just increase the font size and all. Okay. So this seems to be okay. Yeah. So this is the overall course structure. We had already covered introduction and overview of major bioinformatics databases in brief today. Yesterday we covered introduction, and today we've covered overview of major bioinformatics databases, and we are planning to cover subsequent topics, which are bioinformatics applications. Then we are going to do sequence analysis of bioinformatics, along with practical sessions, projects, and question and answer sessions and personal guidances, and we'll be having this collecting and storing sequences. And so basically, alignment of pair of sequences. This what we will do. What we will share this course contents with you all, and we will. So yeah, so we will share it with you, everyone, and I would describe the course fee structure which we are planning to do. This is still under discussion, where we are still evaluating it, and we would want to see the responses of people, and depending on that we might make changes to either the course structure or the fee structure, depending on the way the course structure would be customized or given to a particular person. So basically what we were thinking is our aim. So basically, first of all, the most important thing is to understand is our aim, and our aim is to help the students understand what is bioinformatics as a subject, and also give brief insights into the detailed topics of bioinformatics, to be able to choose bioinformatics as a career for kind of new people, those who have not yet decided on it. So these are the two basic objectives, and there will be certain projects also which will be covered, like drug designing and all, and drug discovery, network studies. Yeah, network studies, drug designing, and all those things would be covered in this current course. Yes, in this course we will cover it. So basically, what we have thought of it as, we'll be putting pricing as $30 a month, and in a month we will be having basically 16 hours of session, and we will be basically providing, we will be doing this for people who will be taking the paid course. They will be able to get the recordings along with live sessions and Q&A on chat, and they'll be able to get the recordings. They'll be able to get the question and answers clarified on email. They will get personal guidance as well from Pragya. And the benefits over here is basically you can pay in installments per month at the beginning of every month. You can check the course contents which we are going to share with you, the Excel file which I showed just now. We will share the PDF version for it, and you can decide to selectively opt for a month having the topic of your interest. And in case you find that these topics, I'm sure these topics are all of them are going to be of great quality, and great preparation would be done by Pragya to conduct them. But at the same time, even if we are doing it the best way we can do it, and still there might be chances that these sessions might not be of interest to you or relevant to you. So you can choose to select the months which you would be opting, the topics which would be covered in the months which you would be opting for. So that also you can do. So this is one way of designing this course which we have done, fee structure and the course which we have done. We are open for feedbacks. In case you have any other feedbacks which you want to give, we are open to basically. Worksheets will be solved at the end of every session. Yeah, so we are going to ensure that you will understand the topics which are covered, so that you worksheets will be there. Yeah, so and we are going to have different worksheets, around 30 worksheets which needs to be solved, so that we want to ensure that you understood whatever is being taught. So 5 projects and the worksheets we're going to include in this whole course. But at the same time, we are also open, and in case someone wants to have guidance on individual topics which requires an hour, 2 hours of sessions, customized topics for a particular fee, and that's it, because the world is moving so fast, and you may not have time to take the full course, and you may not want to wait for the subjects or the topics to be availed in the course. So that's the reason we are open for suggestions. And what we'll be doing is we'll be sharing one Google form link just now within 15-20 minutes, and we will take your interests, and those who are interested in taking up the full course, we would welcome to enroll for the course. We will conduct the course depending on the audience. I think at least if we need to have at least minimum 5 people to start, then we will be starting the course if we get 5 enrollments for the course. And in case there is any custom topics or guidance or something is required, which requires some preparations from our end, and some time and effort from our end, then we will charge on custom basis for that as and when required. If there is a very small doubts and all, then you can always reach out to Bioinformatics Videos Facebook page and post your questions, small questions, that those can be answered by our team, especially Pragya. So that's the whole idea. I will share this fee structure to only those who show the interest in the Google form. To them I will share with this, and once you show the interest, we will share with you the mechanism too for payment as well. So you can pay, and we will ensure that we conduct the course fully, and
We are planning to complete it cost.
yeah, share coursework as an Anu.
Coursework means structure.
Let me do one thing. Let me just unmute others as well. Like that will help. If they want to speak, we can speak. I'll just unmute. Maybe, or maybe I just put a question and put on mute. That's why we have muted ourselves because it was in question answers mode.
So you need to repeat them.
Yeah, so I guess what I wanted to say was that we are almost done with the presentation. In case anyone has got any questions, they can chat, they can put it on the chat, and then we will unmute them, or they can ask on the email. We will share the course structure PDF version on the email with everyone. And make sure you're part of the Bioinformatics videos group. If you're not part of the Bioinformatics videos group, then let us know here on chat or on Facebook videos page, chat, any possible way you can inform us, and then we will, in 5 minutes time, we will conclude the presentation also. We are very, very grateful to all of you for finding out time and attending this session, and we are sure that the session must have added value to your skills and knowledge. And we are very keen to share it with everyone, and that's the reason we have kept it at a very low price point, which is basically not very much looking at today's like scenarios and course structures. You can verify it from any other place, the fee structure on these topics, and it would be definitely much more than at least 30-40% minimum more than this current price which we are offering. Okay, so yeah, thank you so much. We'll wait for 5 minutes. In case anyone is not part of the Bioinformatics videos Google group, they can put in over here so that we can add them to the group, and then we can keep everyone informed about the sessions which we have taken. Okay. Okay. Thank you. Okay. 5 minutes to close. Thank you so much.
Thank you so much, all of you. So I hope you liked the session, and we would like to know that, yes, Upekha, yes, please read, Raj.
Could you please include some information on plant and sample databases in future?
Yes, of course, Upekha. We will see from the plant databases, plant genomes as well. The information, we will take the examples of plant from the plant genomes, the sequences and the accession IDs. And we will take it in the form of a separate project also as research papers. We will make the projects from chosen research papers, and then we will, of course, work on it.
One thing we missed, thanks Pragya for answering that, and one thing we missed to say was that today also in the link which we will share, we will request for feedback because we want to continuously improve ourselves and do better and better and better. So we would request genuine feedbacks. It can be a correction, it can be a suggestion. Request you to be precise on the feedback, like good is all right, I mean that is there, but at the same time if you are able to pinpoint any specific feedback, any correction or improvement, then we'll be very, very grateful to you. And also you can give us a feedback on the pricing point also. What are your suggestions? What do you think? Is it more if you think, or is it less, or is there any other feedback which you want to say on this? Because see, good things are always we have to pay a price for great things any which way. So yeah, so that's the reason we have kept a pricing point. We could have kept it free, but then the value and the seriousness does not come if it is free. So, and we want to ensure that the students learn thoroughly, and that's the reason we wanted to put a price across to it. And also it is about our like no, along with job satisfaction. So yeah, thank you so much. I will share we will share the link for the feedback and the confirmation on the enrollment for the course. Yeah, thank you.